Hello everyone, my topic is post-transcriptional modification. Today we'll be seeing about how mRNA, tRNA and rRNA is processed after its synthesis. The eukaryotic cells have different compartments for transcription and translation. As we know, a transcription takes place in the nucleus and translation takes place in the cytosol. So, these mRNAs that are synthesized in the nucleus must come out into the cytosol before protein synthesis. And there is a large difference between the mRNAs that exist in the nucleus and the mRNAs that are used for making proteins in the cytosol. So, evidently we know that these mRNAs are modified in the nucleus before they can be used for protein biosynthesis in the cytosol. And not only these mRNAs are modified, but tRNAs and rRNAs are also modified in the eukaryotes. So what is post-transcriptional modification? It is also known as co-transcriptional modification, which is a set of biological process that is very common to most eukaryotic cells, where an RNA primary transcript is chemically altered to produce a mature and a functional RNA molecule that can then leave the nucleus and it gets involved in protein synthesis. So what are the modifications that are made after transcription in mRNA? So first one is addition of 5' cap. Second one is polyadenylation, that is addition of poly A tail at 3' end. And third one is RNA splicing. First we'll see what is addition of 5' cap. So this process involves addition of an extra nucleotide at the 5' end of the mRNA. And this newly added nucleotide is also methylated by the addition of methyl group. And also methyl group is added, maybe or may not be. This methyl group is added to second position of OH group of sugar to one or more nucleotides at the 5' end. So this process can addition of methyl group to sugars may happen or may not happen. So in this picture you can see that first one of the 3-phosphate at the 5' end of mRNA is removed. So this process is done by 5' triphosphatase enzyme, and then a guanine nucleotide with its phosphate is added. So the guanine is added by an enzyme known as guanyl transferase enzyme, and methyl groups are added to the position 7 of the base of terminal guanine nucleotide and to the second position of the sugar in the second and third nucleotide. So this addition of methyl group is done by an enzyme called methyl transferase enzyme, and the base on the initial nucleotides also may be nucleated. So if it requires, it methylates the other nucleotides. If not required, it does not methylate. So this is the final product, or 5' capping is done. This is the final product after 5' capping. So you can see that 7-methyl guanosine is at the end, and the other nucleotide is also methylated. So second modification that is done in mRNA is addition of a poly A tail. So this happens in a 3' terminal. So the capping happened at the 5' terminus, and addition of poly A tail happens at the 3' terminus of eukaryotic mRNA molecule. So addition of polyadenosine sequence, that is poly A tail, as many as 50 to 250 nucleotides happens. So these nucleotides actually they are not encoded in the DNA, but they are added after transcription. This process is also known as polyadenylation. And in this picture you can see that first the pre-mRNA is cleaved at position from 11 to 30 nucleotides downstream of an AA-UAA sequence in the 3' untranslated region. Then after cleavage, the addition of adenine nucleotides, that is polyadenylation process, takes place at the 3' end of the pre-mRNA, generating the poly A tail. So in this pre-mRNA processing, a poly A tail is added through cleavage and polyadenylation. So how this is done? This is done by a complex known as polyadenylation protein complex. So it contains 6 factors and it is a multimeric protein complex. What are these factors? So first one is PAP, that is poly A polymerase. Second one is PABP2, that is poly A binding protein. then CPSF, that is cleavage polyadenylation specificity factor, then CSTF, that is cleavage stimulating factor, then CF1 and CF2, that is cleavage factor 1 and 2. So first, the CPSF, that is cleavage polyadenylation specificity factor, binds upstream of UAAA poly A signal 5' end. Then cleavage occurs at 11 to 30 nucleotides downstream of binding site. Then this CSTF, that is cleavage stimulating factor, will interact with the downstream GU sequence and bound with CPSF forms a loop in the RNA. So after this, poly A polymerase will stimulate the cleavage at the poly A site. So basically this poly A polymerase adds approximately 12 adenine residues at a slow rate to the 3' OH group. Now this PABP2 will accelerate this rate of addition of adenine by PAP. After 200 to 250 adenine residues have been added, PABP2 will signal PAP to stop polymerization, and this cleavage factor 1 and 2 are also involved in cleavage process. This is how poly A tail is added at the 3' end. So next, third modification is RNA splicing. In this process, RNA splicing is a process by which newly synthesized pre-mRNA, that is also known as hnRNA, so hnRNA stands for heterogeneous nuclear RNA, is processed and forms a mature and functional mRNA. So hnRNA is processed in the nucleus and it is converted to mRNA, which will then come out to the cytoplasm and it will undergo translation for protein synthesis. In RNA splicing process, the non-coding intervening regions called introns are removed and the coding regions known as exons are joined together. So this process is done by a complex known as spliceosome complex. So what a spliceosome complex contains? So basically the spliceosome complex contains snRNA, which is 5 in number, U1, U2, U4, U5 and U6, and 41 associated protein that adds to its mass, and it also contains 70 other proteins that act as splicing factors and help in binding the spliceosome to RNA substrate and in catalytic process, and another 30 proteins which are presumed to act at the other stages of gene expression. So basically these introns will have 5' splice site, 3' splice site and branch sequence. So these three things are recognized by the spliceosome complex. So these RNA protein complex together forms large splicing apparatus called spliceosome. So what is the mechanism of mRNA splicing? First, the U1 binds to 5' splice site. This U1 will recognize and it will bind to the 5' splice site. Then U2 will recognize and it will bind to 3' splice site. So after binding of U1 and U2, now U4 and U6 will bind together and it forms a complex. So to this complex, U5 will bind and it will become a trimer, and this will come and bind to the intron or branch site. So when all these things are bound to the intron, now this intron will loop out and exons are brought close together. So after this, this 5' splice site is cut. So because of this 5' splice site is cut, U1 will get released, and then the 5' end of intron is connected to the branch site to form a lariat. So because of this, U4 is also released. So by the end of this, U1 and U4 gets released and the intron will become a lariat. After this, the 3' splice site is cut. Then this exon 1 is connected to the exon 2. And this intron, which is in the form of lariat, is released along with other factors, that is U2, U5 and U6. And now the released intron will be degraded. And finally, the exon 1 and exon 2 is connected. This is how the splicing process takes place. So when we are looking into splicing, we also need to understand what is alternative splicing. So this process is also known as differential splicing, where during gene expression, that it allows a single gene to code for multiple proteins. So in this pic you can see in protein A's mRNA all the 5 exons are present, but in protein B's mRNA you can see only 4 exons are present and exon number 3 is absent, and therefore you can see there is a structural change or change in folding of protein A and protein B. And similarly in protein C you can see that only four exons are joined together, where fourth exon is missing. So there is in these 3 proteins, because of this absence of exons, 3 proteins have 3 different structure. So this is how alternative splicing works. So at the end of all the 3 modification, the matured mRNA will contain first the 5' cap and followed by 5' untranslated region, that is 5' UTR, followed by a start codon, that is AUG, and followed by a coding sequence, and after that a stop codon, and followed by a 3' untranslated region, that is 3' UTR, and ends with a poly A tail, So after mRNA, now we'll see how tRNA is processed. So tRNA is processed by removal of a leader sequence and a trailer which is present in the 5' end of the pre-tRNA, and replacement of some nucleotides and modification of certain bases also happens in during the processing of tRNA. So first, for example, you can see in the 3' end, the pre-tRNA contains UUA. So this is changed to CCA sequence in the mature tRNA. And the second thing is addition of methyl and isopentyl groups to the heterocyclic ring of purine base is also done. And methylation of the second position of second OH group in the ribose of any residue. So this methylation will result in mature tRNA, and also conversion of specific uridines to dihydrouridine and pseudouridine also takes place. So that is how the mature tRNA will contain D loop and T size C loop. And some introns is also cut out in this process. So that is how the pre-tRNA is converted to a mature tRNA. So next we'll see how rRNA is processed. So first it contains a 45S molecule. So this 45S molecule is processed to bring 28S, 5.8S and 18S. So how this is done? So basically there are three cleavages takes place. First cleavage occurs at the site 1 and it removes 5' terminal leader sequence, and it will produce two things. One is 41S intermediate and 18S intermediate. So the final 18S is ready. Now this 41S intermediate undergoes second cleavage. So this undergoes second cleavage and it will generate 32S intermediate. Now this 32S intermediate undergoes a final cleavage, that is the third cleavage, and it gets separated to 28S and 5.8S. So finally the processed rRNA will contain 28S, 5.8S and 18S ribosomal RNA. This is how the RNA is processed. So what are the importance of post-transcriptional modification? So protection from ribonucleases degradation, and it stabilizes the mRNA, and lifespan of mRNA is increased, and it also enhances the translation and splicing process, and it also enhances the transportation of RNA from the nucleus to cytoplasm. And modifications, these modifications helps the RNA molecule to be recognized by the molecules that mediates RNA translation into proteins. So I would like to conclude by telling that without post-transcriptional processing, protein synthesis could be significantly slowed down, since it would take longer for translation machinery to recognize the RNA molecules, and also more RNA would have been unnecessarily translated, because in the modification or in the post-transcriptional modification, we will cut off the introns, which is not required, which is a non-coding part. So if this is not there, simply this has to be translated, and which is not necessary for the protein synthesis. So these are my references. Thank you.